Hello, everyone. My name is Marisela Sampaga-Smith, and the title of my project is Every Little Thing She Does is Molting, Using Participatory Science in Improving Data Quality in Indigenous Crab Molt Research. So I completed this project through the Program on the Environment, and I got the privilege to work with Washington Sea Grant's crab team. Specifically, the Molt Search Program is what we specified on. To begin with some background, participatory science, sometimes called community science or citizen science, plays an important role in ecological monitoring because it allows researchers to collect large amounts of environmental data with help from the public. In this case, volunteers submit photographs of Dungeness crab molts or outgrown crab shells that they find on the beach here in Washington, which are then used to monitor crab populations and estimate crab size. However, these submissions are valuable. Inconsistent image quality can reduce the reliability of scientific measurements. So problems such as blurriness, tilted camera angles, poor lighting, or missing scale references make it difficult to accurately analyze the molts. Because of this, my project focused on the question, how can participatory science improve data quality and consistency in Dungeness crab molt research? Or in other words, to be simplified, what are the problems with photo submission and how can we improve or resolve these issues altogether? During my internship with Sea Grant, I evaluated the volunteer submitted crab molt photos using a software program called ImageJ, and I used it to calibrate and measure the carapace width or the spine ends of the crab from end to end, as you see in this photo figure here. I also assessed image quality based on several factors, again, using scale references, blur, lighting, angle, tilt, and specimen visibility. So whether or not the specimen was in frame fully or if it was broken or as a whole in general. I also identified recurring patterns that affected whether a submission could actually be used for scientific analysis for our data, or what ranking could it be qualified as in the hierarchy of data quality. The image here on the bottom left, as you see, shows an example of the calibration and measurement process using the ImageJ. Moving into the results section, one of the main findings was that poor lighting, tilted camera angles, and incomplete specimen visibility frequently reduced my image usability towards the data. Out of all of the issues I observed, however, missing or inconsistent scale references did have the greatest impact on scientific usability. So whether or not they had a standardized tool or a standardized mode of measurement, a lot of them didn't have that included, and that made it a little difficult to calibrate when you don't have a reference to measure. It becomes very difficult and produce dependable size estimates. The chart in the center summarizes 91 flagged submissions and categorizes the severity of blur, tilt, and scale-related issues that I found. While blur and tilt were often minor, scale problems were still more likely to significantly reduce the usability of the image. The photos at the bottom provide a visual comparison between an ideal submission and a not so ideal submission, so a poor quality submission. The image on the left includes proper lighting, visibility, and a clear scale of reference, while the image on the right lacks those quantities and is much harder to analyze accurately. Beyond my direct observations, participatory science literature also supported the idea that improving communication and submission standards can strengthen the quality of community-generated ecological data without reducing the public participation or public involvement part. In other words, better guidance helps volunteers contribute more scientifically useful data while still keeping the projects accessible and inclusive. This leads to my key takeaways here from my project. First, clear instructions do improve submission quality. Second, iterative feedback helps volunteers improve future submissions over time. And third, standardized tools strengthen the scientific value of community-generated data sets. Based on my findings, I suggest that participatory science programs provide clearer photo instructions, standardized scale requirements, and example images of acceptable submissions. Integrating accessible technological tools into the monitoring systems from the get could also help improve consistency and reliability across these data sets. Ultimately, strengthening protocols leads to stronger data, which supports better science and healthier marine ecosystems and overall research for our scientists in the behind the scenes. I also included this quote from a crab team specialist that I found across analyzing these photos, and it was important that they had to have a scale of reference. So it reads, having an item for scale helps us make the most of your reports and keeps our data quality high. And this was in response to a photo submitted through a volunteer, and that photo feedback was valuable in the volunteer's next submission. So it was well received. Finally, I would like to acknowledge my site supervisors, Lisa and Hannah, the Washington Sea Grant Crab Team, and all of the volunteers and participants who were involved in the research process as a whole. Thank you so much.